All right. So yeah, I would like to start by thanking the invitation. It's a pleasure to be here and talk a little bit about our research, which actually started, and most of the work was done back when I was a postdoc in the Gladyshev lab back in Harvard. And now I'm an assistant researcher in Lugarinho's lab here in Portugal, my home country. And so, I'm going to talk a little bit about how aged clonal B cells can drive cancer. So I will just start by a brief introduction, and I don't think I have to convince this audience that aging is the main risk factor for chronic diseases. And here it's a nice study from Lancet in 2017, and you can clearly see that with age there's a dramatic increase in many of these chronic diseases, such as, and I'm just highlighting here, these are the most prevalent ones, and you can see that cancers and cardiovascular diseases, both in males and females, they dramatically increase with age. So, but what is it with aging that drives these chronic diseases? And this is one of the questions we are interested in. And I highlighted that aging is the main risk factor for cancer, for example. And it's interesting because currently there are no good models to study age-related cancer. Most of them rely on transgenics or knockouts or even chemical-induced models to drive cancer. But in my opinion, I think this fails to recapitulate how for example, aging microenvironment helps to initiate and drive cancer. And if you look at, and this is why this whole started actually, if you look at rodent animals that we used in the lab, and here it's just showing black 6, but it's mostly true for many other strains, you can see that lymphoma or B-cell lymphoma is the most prevalent cause of death in these animals. And so we kind of set up to study how aging actually promotes cancer using these aged animals. And so, we started with a very straightforward, very simple experimental setting where we took young animals and we compared to old animals not carrying any lymphoma and old animals, age matched, of course, carrying B-cell lymphoma. And I have to tell you that this came from a collaboration with Harvard Rodent Pathology Core that assessed the cause of death after we did all the analysis. So this was completely unbiased. And so we took the spleens to extract B cells, because we know that the spleen is the one of the most, if not the most important place where B-cell lymphoma actually starts. And we did a bunch of sequencing like RNA-seq, RRBS for DNA methylation clocks that I won't spend much time today on this, but we did also immunoseq to assess clonality. So this is sequencing of the CDR3 region common in the BCR receptor in B cells. So this gives an idea of the clonal level of a certain B cell population. And we also did whole genome sequencing, whole exome sequencing, and proteomics, which I won't spend much time on. And so with all of this, we expected to answer the questions: what are the mechanisms that might be driving these B cell changes with age that might lead to cancer? And so, I'm going to show you some simple facts that kind of was the kickoff of this project. We found, and this was kind of surprising, that in old animals we found B cells to increase, but this was most dramatic in lymphoma cases. But of course, this was a total B cell population, so we had to go deeper into this uncoupling of this B cell population, and we ran the usual populations using antibody panels, and we found we look at follicular B cells, germinal cells, marginal cells, and what give you, and it was nice to verify with a common hallmark of the immune system that follicular cells, they drop with aging, but most dramatically in these lymphoma cases. And accompanying these changes, we found this mysterious double population, which is that was double negative for CD21 and CD23. And of course, at this point, we could tell if this was just one population or two populations or even three populations. We just found that these double negative cells were increasing with age. And I'll come back to this a little bit later. Yeah, and so a little bit more of a characterization. We found that lymphoma cases, age-related lymphoma cases, they have spleen weights that are larger, heavier than controls and even old animals. And that and this was an interesting finding that the average mean of each B cell size, it's increasing with age, and especially with in lymphoma cases. And so this is kind of a nice marker that we could verify over and over that animals with B-cell lymphoma had overall average cell size increase with aging, but most dramatically with lymphoma cases. And then this is just to tell you that with immunoseq, this what I just told you like a few minutes ago was used to assess clonality. And you can see that there's, increased clonality in lymphoma cases. And this is what you would expect if you have cancer, because we know that clonality is prevalent in cancer. There's some clonals that get, selected and gets, overpopulates the tissue. And this is exactly what we found. And using RNA-seq, looking for reads for Ig diversity, we see that in lymphoma cases, there's a drop in the diversity. So it nicely matches two different assays showing the same thing. And again, with whole exome sequencing, looking to variant allele frequency, we see the same. So lymphoma cases are extremely clonal. And just the bottom line of this slide, what I'm telling you is that young animals have a diversity of B cells that with age, some of them get larger and start getting selected. And in the case of lymphoma, you get one particular clone that gets selected and the cells are usually larger. Okay, and so this was, we continue on characterizing these B cells, and we found something surprising. We know that usually lymphoma B cells, they have isotype switching, but we didn't find this in our lymphoma cases. We found that these lymphoma B cells are mostly IgM positive. So this suggests that they didn't undergo germinal center reorganization, rearrangements, and if you look at mutations in the CDR3 region using the immunoseq, we found that the top clones, so B cells that were samples that were highly clonal, do not go under this somatic hypermutation. And together, this suggests a non-canonical way of B cells being transformed into lymphoma. And, this is just a teaser. We did more characterization, but I just wanted you to keep in mind that whatever this process is, it doesn't seem to be a canonical pathway driving lymphoma. Yeah. No. Okay. And so if you do some PCA analysis for transcriptome, promoter, DNA methylation, proteome, whatever, you find that samples from lymphoma B cells, they cluster away in any of the different assays we tried. They cluster away from young animals, and this was expected, but very different from old B cells that cluster much together with young animals. And this tells us that this clearly reveals that these B cells from lymphoma, they have distinct molecular patterns. And so obviously the next question was, what are the molecular mechanisms that might be driving this B cell age-related transformation? And to answer that, we used this sequencing data. We did gene set enrichment analysis. And what this plot shows is young versus old for methylation, young versus old for RNA-seq, and the same for the comparison of old versus lymphoma. And you can nicely appreciate that with age, you get turned on many of the common pathways, for example, mTOR signaling and inflammatory pathways, interferon gamma. This is often observed in aging signatures. But when the animals transition into a B-cell lymphoma, you get these pathways even more further increased, and you start seeing, for example, increase in MYC targets, very, very highly expressed. And what is also interesting was to realize that these inflammatory pathways, intracellular, they seem to get shut down. So whatever these cancer B cells are having, they seem to become insensitive to the microenvironment, and this is something we are also looking at the moment. Yeah, and so I told you that the cells are larger, they have this MYC target, so we decided to study a little bit more into detail this MYC, and we found that, for example, genes under the regulation of MYC, they are upregulated mostly in lymphoma cases. And so what is driving the activity or the overexpression of MYC, we didn't know. And when we submitted initially the paper, the reviewers say, okay, this might be nice, but what if this is just a translocation, right? Because many of the lymphoma in humans, they result from translocations in the MYC gene. So, you know, to answer this question, and sometimes the reviewers can be helpful, and this is actually one of those examples. We did whole genome sequencing comparing old B cells from lymphoma B cells, and to keep this short, this is all the chromosomes, and then you have specific, let me just zoom in to show you. For example, these black boxes, they refer to duplications in the genome. When it's red boxes, they refer to deletions. And when we look at these lines, they refer to translocations, either in lymphoma B cells in red, or just old regular B cells in blue. And these green boxes, they are genes that are often seen to be translocated or duplicated in lymphoma cases. And I highlighted here MYC to show you that there's no duplication or deletion or even translocation in the genomic region for MYC. So to sum up, this, whatever is activating MYC and driving its overexpression, it's not caused by a translocation or duplication. It has to be something else. And I'll come back to that later. So yeah, and so taking a few steps back, I showed you that we found this double negative population to emerge with aging, and most specifically with lymphoma. So we set up to try to identify what this population was. And actually, we're lucky to stumble upon this publication by Kimmel, where they did some single cell sequencing for many tissues, comparing old versus young. And they were actually showing two populations emerging with age, and they described this as aging manifests novel neoplastic B cell states in the spleen. So we thought that maybe we had hit the jackpot. We, decided to look a little bit into detail into this population, and we found it to be double negative. So these two populations, they were double negative. And if you look at the enrichment scores that they were describing in this paper, you find many of the same pathways that I just showed you a couple of slides ago. We have MYC targets overexpression, mTOR, even oxidative phosphorylation that I didn't highlight it, but we also have it. And then you have the shutdown or inflammatory pathways. So the gene expression profile was very similar to the one I was showing you before. And so we decided to look into this, but we did something else. We combined this Kimmel data set with tabula muricenis, and we found that this data set from TMS, tabula muricenis, they also had the double negative population. We integrated the data and we got the question, what is our population? So to answer this question, we used the clonality signature, which is not more than correlating overexpressed genes with clonal cells. So the more clonal cells, B cells we have, we looked into their gene expression and we did like a correlation to derive this clonality signature. And this population here was highly clonal. This one turned out to be ABC cells. So these are age-associated B cells. They are overexpressing TBAT. This is already described population. They are involved in chronic infections and autoimmunity, which is interesting. But the population we were, we couldn't find a name for this population, so we named it as age clonal B cell population. And these are comparing, they are similar, they are very similar, but one of the differences is that they are enriched in MYC targets comparing to this population was mostly enriched in TBAT, which is a transcription factor for these B cells. And because the gene expression was similar, except for this one that I just told you about, we wonder if this ACBC population, these clonal B cells, would emerge from ABC. So to answer this, we resorted to pseudotime analysis, which is based on gene expression, only to find that ABC cells, they derived from follicular B cells, and this is kind of known from the literature. So, this is kind of reassuring to see that at least at this part, this is working. And then it predicted that ACB cells are arising from these ABC cells. Okay, so we think that these clonal B cells, they emerge from some kind of modification that may happen in these ABC cells, that I'm going to show you something about this in a couple of slides. But then we went to the mouse, because this was just in silico, and we went to the mouse just to validate if this was the case, and we validated through the panel of of proteins that we found to be unique for each population I just showed. And using the follicular B cells panel, we again show, like previously, that follicular cells, they decrease with aging, but most dramatic in lymphoma cases, that these ABC cells, they emerge in old animals, but not in lymphoma. And if you look at ACB cells, this clonal population, they dramatically increase their numbers in lymphoma cases. There's a slight increase with aging, but it's most dramatically observed in lymphoma cases. And so this tells us that this is not just an artifact from computation. We could actually isolate these cells from the animals. But then the question, how do these ACBC cells arise, came up. And to answer this, we resorted to CellChat, a software that predicts, based on gene expression, interaction between cell populations. And so the thicker the line, the more expected, expected predicted interaction between two cell populations, sorry. And you see that ABC, they are predicted to interact with follicular cells and ACB cells, but they mostly interact with themselves. So we wondered if it's this interaction between two ABC types that leads to one becoming clonal. So the idea is that they might interact, and one of these cells, because they might have, for example, a certain type of somatic mutation, gets selected and starts overexpressing MYC, and then overpopulates the tissue. But what could we use to test this? And the tool also gives you possible top hits. And we found, for example, if you look at ABC cells profile, both outgoing signalings and incoming signaling, we found that CD22 was highly showing up as a top hit. So it's actually interesting, because after doing some digging, we found that CD22 is upstream of c-MYC activation that may lead to cell growth and cell cycle. And so, we devised an experiment co-incubating ABC cells with follicular B cells. This is not the ideal experiment. So ideal, we should have two ABC cells labeled differently and then co-incubating, but we couldn't do this. We couldn't perform this experiment. So we just incubated ABC with follicular cells and assessed whether this would lead to a cell size increase and c-MYC overexpression. And of course, this in the presence of CD22 would be expected to reduce the cell size increase in follicular B cells. And so this is follicular B cells incubated just with the antibody. This is the normal size for follicular B cells. If you co-incubate follicular B cells with ABC, bam, you have an increase in follicular cells B cell size. But most interesting, if you co-incubate with the antibody, you blunt the cell size increase and also c-Myc overexpression, which I'm not showing just for the sake of simplicity. But so, so this kind of tells us that c-Myc overexpression is cell size that helps the certain clone to get prevalence might be coming from this interaction through CD22 signaling that leads to c-Myc overexpression. Yeah, and another thing we wanted to ask is whether these ACB cells have an impact on lifespan. Can they reduce lifespan? So initially, we kind of started a longitudinal setup, collecting blood regularly until mice were dead. And long story short, we found that animals that early accumulated ACB cells, they tend to die two months earlier than animals that do not accumulated ACB cells. And this is kind of the pictorial representation. So animals with early ACB cells accumulated, they die around 2 months before animals that do not accumulate these ABC cells, which, this was very interesting, but again, the reviewers said this is, association data. So you need to show a causal effect. So in order to do that, we prepared regular size spleen extracts from aged animals without lymphoma, and we did the same from animals that had lymphoma. We collected the extract and we injected in REG2 knockout mice. These are a good model. These are young mice, but these are a good model because they do not develop mature B cells or T cells. And so whatever you find is because we injected and not because mature B cells are maturing or T cells are maturing. So, and we looked at lifespan data, of course. And so this is just to show you that after a while, you do find ACBC cells in the spleen after the injection. So, and this has to be from our injections, because as I just told you, they do not maturate B cells or T cells, so they do not migrate into to the spleen, for example. And this is very nice to see that animals injected with these lymphoma extracts containing mostly ACB cells, they start dying after around 1 month of injections. The controls are still alive. Negative controls, just we let them age, or the old or animals injected with old extracts not containing ACB cells. And so I think this is clearly shows that the potential of these cells to lead to shorter lifespan. But of course, then now we're kind of expanding our findings, and we decided to look if these ACBC cells, they invade other tissues. And so, I won't go into much of a detail, but we used this data set published in Nature from Sciinov Group in Stanford, and we found that so each dot is a cell, and the yellow word the cell is, meaning that they have our ACBC cell signature. And we found that many tissues, they accumulate these B cells with our ACBC cell signature, especially kidney, liver, and lung. So these are peripheral tissues. And so we just went back to the lab and tried to recapitulate this. And we did some stainings. And these are just animals without lymphoma, and their tissues, liver, lung, and kidney. And if you look at animals with lymphoma, you find this abnormal accumulation of B cells. And of course, this doesn't tell you that these are ACB cells. We need to prove that with single cell sequencing we're planning to do. But, it points that there's definitely infiltration of abnormal B cells in this age-related lymphoma. And so we think that this might be our clonal B cells. And just to wrap up this presentation, why is this all any relevant? Well, if you look at our signatures in humans using GTEx data for spleen, we kind of divided people that had low IG diversity, and we assigned them as clonal B cell individuals. And then we projected at our signatures, and you find that ACBC signature is nicely correlating in these individuals, but not in people aging without clonal B cells. We didn't see in clonal T cells, so it seems to be specific. And for example, if you look with individuals without any clonal B cells, you get the ABC phenotype, but not the ACBC phenotype. So this really illustrates that with age, you have ABC cells that may later lead into ACB cells if the cells become clonal. And in collaboration with Retro Biosciences, Rico has been doing great work together with us. And he looked into a data set with blood from humans. And here you see the compilation of all these individuals ranging from 25 years old until 65 years old. And you find that there's this emergent population of B cells with age. And if you used our signatures, you see that they are enriched mostly in ABC cells here, same place, ABC cells. And if you run MIC targets, here they are. So we really think that this might be a biomarker of age driving B cell size, B cell lymphoma in humans as well. And if you look at clonality scores, because these guys also had BCR sequencing data, we find that only this population is highly clonal. So this completely fits with our mouse data, and we think we might be dealing with some kind of new population that might be used for as a biomarker. Yeah, and of course, then we wanted to see how this would recapitulate in terms of transcriptome, DNA methylome, and is even somatic mutation. So we compare this with many different lymphoma cancers, also non-lymphoma cases such as esophageal carcinoma. And we only see specificity like recapitulation in human blood lymphomas, and most specifically diffuse large B-cell lymphoma and follicular B-cell lymphoma, if you assemble all these three analyses. So whether this is really B-cell, these two types of lymphoma, we don't know, but it might be related to a more general mechanism of transformation of age B-cells into cancer. And so we're digging into this a little bit more into detail now. And this is the running model. We are, as I told you, we are under review. We are about to resubmit the paper. So our model suggests that follicular B cells with age, they become transformed in ABC cells. They start interacting with themselves. They drive MYC overexpression. And I didn't show you this pro-inflammatory, but these cells seem to be secreting and contributing to inflammatory microenvironment that will infiltrate other tissues and cause tissue damage. But this is another project we are actually starting now. Yeah, so, and I didn't have time to show you, but we did rapamycin treatments, we did CMYC deficient animals, and this seems to block this transition from ABC to MYC cells. And so this seems to be dependent on mTOR, on MYC, and also CD22 interaction as well. Right, so I'd just like to finish by thanking amazing people that contributed to this study. Vadim, which it was my mentor of this study. I had a great time in Boston. Nastya, which is the co-first author of this paper, which is an amazing friend, and we keep talking almost every day. Alexander Tishkovsky, which is helping with biological age assessment of these cells, which I didn't have time to show. Albert was helping with single cell, Jabba with DNA methylation that I didn't have time to show, and the whole lab, of course. And also my fellowship that allowed me to go to Harvard, National Institute of Aging, and all these collaborators that helped this work moving forward. So to them, my big thank you. Yeah, so basically that's it.